Okay, welcome to the lab on the urinary system. We're going to be looking at some structures from the models that are available in lab, but we'll also take a glance at some of the fetal pig structures, even though we don't have fetal pigs in the lab this week that I can see. We'll take a peek at those structures. And this is just a list of what we need to know and what we want to go over. Okay, so make sure and take a note of that as you look at the downloadable notes off of Angel. So, the urinary system's job is to clean your blood, to maintain fluid and electrolyte balance, and um, essentially to uh, get rid of nitrogenous waste. That's one of its most important jobs. The 12th rib is the last rib that's attached to the vertebral column, and it's a floating rib um, due to its single attachment. And you can see that the two floating ribs protect about the upper two-thirds of the kidneys, which are the blood cleaning organs. And because the kidneys are so close to the posterior body wall, a blow to that region could easily dislodge one of these organs. That could occlude the flow of urine from the kidney down to the bladder, and as a result, those types of blows are disallowed in wrestling and boxing and ultimate fighting and so on. They're called kidney punches, and they're illegal because they're extremely dangerous. Not only can you dislodge the organ, but if you dislodge a floating rib, it could easily puncture not just a kidney, but any of the other organs that are located in the abdominal pelvic cavity. So this is just a look at the urinary system. And the kidneys are about the size of a bar of soap, and they clean all the blood in your body every day. The renal arteries deliver the blood to the kidneys, and the renal veins drain them. The ureters deliver urine to the urinary bladder, and then the bladder empties the urine through the urethra. And so you can see here, um, you've got one kidney on either side. The right kidney sits lower than the left because of the liver on top of it. The adrenal glands sit on top of each kidney. This is the renal artery here, and these are the renal veins that drain the now cleaned blood back into the inferior vena cava, where it'll eventually head back up to the heart and be pumped throughout the body. The ureters, these structures here, two very thin tubes that conduct the urine down here to the bladder. The ureters and the bladder are lined with a special type of epithelium called transitional epithelium. It has the unique property of being able to stretch when it fills up with fluid. So fairly important tissue feature here. If we don't clean your blood every day, if we don't remove the nitrogenous wastes and the toxins, what would happen is it would begin to build up inside the tissue fluid and the blood plasma, and it would begin to change pH. Most nitrogen-containing compounds are bases, so they're going to cause the pH of the solution that they're in to rise. And remember that if pH rises too much, we run the risk of changing the voltage around cell membranes, so perturbing membrane potential, and also possibly denaturing enzymes, and that would cause their chemical reactions to shut down. And as a result, you would die. So if your kidneys fail and you don't have any other way of cleaning the blood, this leads to a condition called azotemia, which is the buildup of nitrogen-containing waste in the blood, leading to uremia, which is the buildup of urine in the blood, which can lead to coma and death. This is why in people that undergo acute renal failure, we have to clean their blood artificially using dialysis. And here you can see the abdominal pelvic region of the torso model. Notice the different structures here. The spleen, there's the renal artery and vein, there's the kidney. Okay, that's the right kidney. Remember, this is the pancreas, this is the duodenum. That is the right ureter, that is the left ureter, and down there is the urinary bladder. And this is a male torso. We know this because this is a transverse section taken through the penis. And in men, as we're going to find out, the urethra runs essentially from inside the pelvic cavity and out of the body through the penis. It has three regions, one that runs through the prostate gland, which is on the underside of the bladder, called the prostatic urethra, one called the membranous urethra, which is essentially contained in the region that transitions from the abdominal pelvic cavity to the outside world, and then the penile or spongy urethra, which is encased inside the glands in the shaft of the penis. You'll also notice, if you can take a sort of a detailed look down here, that you've got three columns of tissue that run through the penis. The two upper columns are called the corpora cavernosa, and the lower column of tissue is called the corpora spongiosum, and their purpose is to allow erection to occur during sexual arousal. What happens during an erection is that the rate of blood flow into these columns of tissue is greater than the rate of blood flow out, and as a result, the organ becomes erect and capable of intromission. Here you can see the structures in the pig. Okay, the kidneys in the pig are rather large. You can see here the ureter, the kidneys. Okay, down here you can see the bladder in the pig is kind of oblong. In our model for the human abdominal pelvic region, it's shaped more like an oval sac. But remember, it has the ability to stretch, to distend. That's one of its major purposes. You can see here just another look at the abdominal pelvic region. Here you can see the rectum filled with fecal material. Okay, you can pick up here ureter. There's one ureter. There's the other ureter. There's a kidney. Here's another kidney. And you can see the equivalent structures here in the model. Okay, notice that in our model here, the rectum goes behind the bladder, okay, as it does in the pig, and of course it empties into the anus, and that's the passage that fecal material takes out of the body. This is just a look at the structures in the female fetal pig. Slightly different arrangement of the urogenital system in the female. The urethra is much shorter, and of course the gonads are inside the body instead of outside. And you can see here the bladder, okay, indicated here in yellow. You can see the ovaries here in pink. These structures here are the fallopian tubes, okay. They conduct the egg from the ovary into the uterus. The structure that's medial to these is the uterus here, lies right between the two uterine horns, and then this leads into the vagina, okay, which leads out of the body. The bladder, okay, empties into the vagina, and there is a urethral opening that dumps straight into the urogenital sinus. So in the pig, we have a single opening for the vagina and the urethra that exits out through the body. In the human being, they're two separate openings. Okay, so you can see here again the urethra, and down here the urogenital sinus, which is where the vagina and the urethra intersect. In the male, okay, you can see the urogenital structures here, the bladder up top, the penis, okay, and down here you've got the bladder, and back here you have the urethra. Okay, remember that in the male, the urethra is the passageway for both semen and urine, so it's a common opening to the exterior. Okay, here you can see again same view, but you can pick up now in our inset in the model, you can see the penis, and you can see the urethra, which is going to be right in the middle of the corpora spongiosum on the ventral side of the penis. There's the bladder, the ureters, the kidneys, of course, the adrenal glands. Okay, one of the things that we want to understand about the kidney is that its primary job is to serve as a filter. So filtrate is made by the nephrons, which are the filtration units of the kidney. You've got about 1 million in each kidney. The filtrate that is derived from the blood contains water, ions, glucose, and amino acids um, up through the region of the nephron called the proximal convoluted tubule. And the way that the fluid flows through the nephron is it runs from the renal cortex to the medulla, and then it runs back to the cortex, and then it runs down to the medulla one more time, and it empties into the calyces. In the calyces, the filtrate is considered to be urine. Urine then flows from the calyces to the renal pelvis to the ureter, then to the urinary bladder, then exits the body through the urethra. And so what we're looking at here in this inset is a picture of the kidney. The kidney has several parts. Okay, it has an outer capsule, which you can see here, and it has a layer of tissue underneath the capsule called the cortex. Okay, this is all cortex here, as are the regions here between what we call the pyramids. These are part of the cortex called the renal columns. And then the medulla is populated by these triangular structures that are called renal pyramids. You can see these structures in the model as well. Okay, you can pick up the capsule. Okay, going all the way around. There is the medulla. And then, of course, this is the capsule, this is the cortex, and then these pyramids make up the medulla. Those are the calyces. The first calyx that the pyramid empties its contents into is called the minor calyx, and these converge into a larger cup-shaped structure called the major calyx, which converges onto the renal pelvis, which dumps into the ureter, and that's the path of urine. What's indicated here by the pyramid and its overlying cortex is what we call a renal lobe. Okay, and the way that blood flows into the kidney, the pathway that it takes before it's about to be cleaned, is it heads into the kidney through the renal artery. The renal artery then breaks up into segmental arteries, which break up into interlobar arteries that go between the pyramids. These then arch around the base of the pyramid, forming the arcuate arteries. And then off of the arcuate arteries, which I'm touching here, okay, are these very tiny arteries that are called the cortical radiate arteries, and some textbooks will refer to them as the interlobular arteries. What happens next is that there are even tinier branches that come off the interlobular arteries called afferent arterioles, and these very tiny arteries lead directly into a cluster of capillaries called the glomerulus, which is enclosed by a structure called the Bowman's capsule, similar to the way that a softball would fit into a softball glove if the glove were the capsule and the softball were the glomerulus. Okay, if you can picture that. And it's inside the Bowman's capsule that filtration of the blood occurs. The filtrate then moves through the regions of the nephron, and what happens in the nephron is that we adjust the chemical content of the filtrate until all that's left by the time we go all the way through the nephron are waste products, water, some electrolytes, occasionally some hormones, and that's pretty much it. Okay, that's really all you should see in normal urine. And so what you've got here with the color coding is just a way for you to orient yourself in terms of where the blood flows. Now, once the blood has been cleaned, after it's been filtered, and after there's been some exchange of waste products and nutrients between the blood and the filtrate that's in the nephrons, the blood will then exit the kidney. It'll go from the peritubular capillaries, which surround the nephron, to the interlobular veins, to the arcuate veins, to the interlobar veins, and then ultimately empty into the renal vein, and from the renal vein will empty into the inferior vena cava. And by the time the blood reaches that point, it's been completely cleaned. All the nitrogenous waste has been removed from the blood, and we're able to stay happy and healthy for another day. Okay, so this is just showing you the different parts of the kidney. This kidney is actually upside down. Okay, this is the adrenal gland, which is on top of the kidney. So this is actually the top, and this is the bottom. And you can see here the ureter, the renal pelvis, and so on. Okay. Here you can see some of the other gross structures of the kidney. We sort of have to reorganize this a little bit. Hold on a second while I fix this diagram. Okay, now you can see the kidney the way it's supposed to be laid out. There's the pelvis, there's the minor calyx, there's the major calyx, there's the ureter, there's the adrenal gland. Okay. Now, what you're looking at here is a close-up of the area where the cortex and the medulla touch each other. Okay, the medulla starts with the base of the pyramid and heads towards the apex of the pyramid that's proximal to the minor calyx, and the cortex is that granular tissue that overlies it, and then superficial to the cortex is the capsule. So the renal cortex contains the glomeruli, and they are of course enclosed inside the Bowman's capsule. The cortex also contains the distal and proximal convoluted tubules, and this is why if you dissect a kidney and look at the cortex, what you find is that it has a rather granular appearance compared to the renal pyramids, which appear to have longitudinal stripes running down them. And the reason that the medulla appears striped and the cortex appears granular is due to the fact that the pyramid has parallel arrangements of loops of Henle, collecting ducts, and peritubular capillaries that all run parallel to each other. The glomerular capsule contains the glomerulus, which can be found here in this drawing. You can see a couple of glomeruli here. You can also see them in the lab under the slide. We'll take a look at some kidney tissue under a slide. And you can also see the distal and proximal convoluted tubule. The renal medulla contains the loops of Henle and the collecting ducts. The collecting ducts empty into the calyces, which empty into the renal pelvis, and the glomerulus and the tubules are part of the nephron. Okay, so we, in the next slide, break down the different parts of the nephron. You can see them here. This is a zoom up of the kidney where the cortex meets the medulla, and what we have to understand is the flow of filtrate through a nephron. Okay, so what I'll do here is I'll trace a nephron for you. This is a Bowman's capsule. This is the PCT. Okay, this is the descending limb of the loop of Henle, followed by the ascending limb, followed by the distal convoluted tubule, or DCT, and then this empties into this structure, which is the collecting duct. Okay, so that's the flow of urine here. Okay, the flow of what starts out as filtrate and which by the time it gets to the collecting duct is considered to be urine. Now, there are different tasks that take place in different parts of the nephron, uh, which contribute to the task of cleaning the blood and eliminating the waste products. When the blood's initially filtered in the glomerulus, you can see right here, what happens is that any molecule below a certain molecular weight is going to pass into the filtrate. That includes useful solutes and also stuff that we want to get rid of. So you'll see amino acids, you'll see glucose, you'll see fatty acids, um, and you'll also see waste products like urea and ammonia, creatinine, as well as a lot of water and electrolytes, all enter the filtrate through the glomerulus into the Bowman's capsule and into the proximal convoluted tubule. But once we're in the proximal convoluted tubule, what will happen is that through active transport, we'll rescue most of the useful solutes out of the filtrate and we'll put them back into the bloodstream. So by the time the filtrate leaves the PCT, it shouldn't have any amino acids, no glucose, no fatty acids, and a significant amount of the water and electrolytes should also have been reabsorbed back into the bloodstream. And the vessels that do the reabsorbing are called the peritubular capillaries. They get that name because they surround the kidney tubules. Now, as the filtrate descends into the medulla, what happens is that a significant amount of water leaves the filtrate, and so the filtrate becomes very concentrated. The salt content gets up to about 1200 milliosmolar by the time we get to the bend in the loop of Henle. And the reason that that happens is because the medulla of the kidney has a salt gradient in it. It's very salty compared to the fluid that's in the tubule. And so what happens is the water leaves through the descending limb of the loop of Henle by osmosis because the descending limb is water permeable. Now when we get through the bend in the loop, what happens is that the ascending limb of the loop of Henle changes its game and it actively pumps sodium and chloride into the medulla of the kidney, and the purpose of this is to establish the salt gradient in the medulla. By the time the filtrate reaches the distal convoluted tubule as it comes out of the ascending limb, the osmolarity is the same as it was when it left the proximal convoluted tubule, or very close. And so what you might wonder is, well, what was the point of that? We made the filtrate very salty, and then we turned around and we made the filtrate very unsalty. Well, the point of that was to establish the salt gradient in the medulla, because what happens in the distal convoluted tubule is that significant amounts of waste products are excreted into the filtrate. A lot of water is reabsorbed in the cortex, and then the filtrate moves into the collecting duct, and it's this last trip through the collecting duct that dumps down through the medulla that's going to determine the water content of the urine. And the permeability of the collecting duct of water is governed by a hormone that's made by the posterior pituitary gland called antidiuretic hormone, or ADH. It also sometimes goes by the name of vasopressin. When ADH is present in high amounts, what happens is that the collecting duct is extremely permeable to water. And as a result, what happens is that most of the water in the urine goes back into the kidney, and from the kidney it goes back into the bloodstream. When this happens, the urine that's produced is very concentrated, so it'll have a yellow to almost orange appearance. When ADH isn't being made in high amounts, the collecting duct becomes relatively impermeable to water. A lot of the water stays in the urine, and the urine takes on a pale yellow to almost clear appearance. The purpose of this is to regulate your body's fluid balance and to control your body's blood volume and blood pressure. So ADH is secreted in response to a drop in blood pressure or a drop in blood volume. Remember that diuretics are drugs that make you urinate, so an antidiuretic is going to be a compound that causes you to hold on to your body water. And the whole reason that the collecting duct can do this is because the medulla has a salt gradient in it, and so water is allowed to leave by osmosis when the collecting duct is rendered water permeable by the action of ADH. So that's the whole point of what the loop of Henle was trying to accomplish. What you have to remember when you look at the nephron is that it's not a uniform tube from its start to its finish. The cells that are in different sections do different jobs because they have different kinds of pumps and channels on their surface. The proximal convoluted tubule has pumps and channels designed primarily to grab useful solutes out of the filtrate and put it back into the blood. The descending limb of the loop of Henle is mostly a water permeable set of cells that don't have a lot of pumps in them, so most of the transport that occurs there is passive. The ascending limb has a lot of sodium and chloride pumps that actively move sodium out of the filtrate and into the kidney. And then finally, in the distal convoluted tubule, we feature a lot of pumps and transport mechanisms that secrete waste products into the filtrate before it enters the collecting duct. And then the collecting duct is simply a tube that will either be permeable or impermeable to water depending on the action of ADH on the cells that line the collecting duct. Now, this salt gradient that's so critical to maintaining our water balance is actually established by the juxtamedullary nephrons, and you can tell juxtamedullary nephrons because their loops of Henle are very long. They extend almost to the apex of the pyramid, in each pyramid in the renal medulla in the kidney. The rest of the nephrons, the other 85%, are primarily blood detoxifiers, and the bulk of their structure is in the cortex. But they have to work together in order to clean the blood and maintain fluid balance. Just because the juxtamedullary nephrons aren't present in nearly as high a percentage doesn't mean that they're unimportant. Now what you're looking at in this slide is just a flow of blood through the kidney, and it simply illustrates what we were talking about earlier. 1% of the filtrate passes out of the collecting duct as urine. The other 99% is going to end up back in the blood as water. If this didn't happen, we would dehydrate very rapidly. Our fluid balance would become dysregulated and we could very easily die. So filtration occurs at the glomerulus. You filter about 90 2-liter bottles of water a day through the kidneys. That's about 180 liters. So think about that the next time you go to the grocery store and look at a 2-liter bottle of pop. Okay, imagine 90 of those going through two structures the size of a bar of soap every day. That's a lot of work for such a little organ. So if we look at the flow of blood, you can see that we run from the aorta to the renal artery, to the segmentals, to the interlobar arteries, to the arcuates, to the cortical radiates, which again are sometimes called the interlobulars, to the afferent arteriole. Then in the glomerulus we do the filtration of the blood. Then that blood flows into the efferent arterioles, which then flow into the peritubular capillaries. So the way I always remember that, E is for exit, okay, so efferent is the exit from the glomerulus, so that's how you're able to tell the afferent from the efferent. The other thing that you'll notice when you look at the models or look at the diagrams is that the afferent arteriole always has a bigger diameter than the efferent, and the purpose of that in part is to maintain a certain blood pressure inside the glomerulus. The efferent arteriole is going to empty into the peritubular capillaries, which surround the loops of Henle and the distal and proximal convoluted tubules. These will empty into the cortical radiate veins, also sometimes called the interlobular veins, which dump into the arcuate veins, which dump into the interlobar veins, which empty into the renal vein, which is going to empty into the inferior vena cava. And that's the whole trip. And if you're wondering if you have to know it, yes you do. What you're looking at here is just the structure of the glomerulus. This is the filtration apparatus that starts the process of cleaning the blood that flows through our body. Remember, you've got 1 million of these per kidney, and you can see that the blood flows in through the afferent arteriole, goes into the glomerulus, and then heads out through the efferent arteriole. And there's a few features that we should make note of. One is what we call the urinary pole. The urinary pole is where the Bowman's capsule leads directly into the proximal convoluted tubule, or PCT. The afferent arteriole is the pathway that blood takes into the glomerulus. The efferent arteriole is the exit from the glomerulus that leads to the peritubular capillaries. And what happens in the glomerulus is filtration. And there are 3 layers of filtration that the solutes have to go through, the solutes in the water have to go through in order to get into the Bowman's capsule. The most superficial are the podocytes. Okay, you can see these weird looking endothelial cells that kind of wrap their extensions around the capillaries, and they make little filtration slits that only allow things below a certain size to get through. And then there's a basement membrane that is underneath the podocytes. And then you've got endothelial cells that line the capillary, and the endothelial cells are in direct contact with the blood. So you have to get through all 3 of these membrane layers in order to get into the glomerulus. And so if you're above a certain size, you're going to stay in the blood. And so you should never see in normal blood any of the formed elements. You should also not see protein because it's too large to get through the filtration apparatus. And if you do, that indicates that we have damage somewhere along the urinary system. Okay, it could be damage in the glomerulus, it could be damage in the nephron, it could be damage in the kidney, in the calyces, in the ureters, or even in the bladder. Okay, that can cause things to appear in the urine that shouldn't be there. Now, the opposite pole from the urinary pole is called the vascular pole, and it's named that because this is where the blood vessels go in and out of the glomerulus. And you've noticed that the afferent arterial diameter is bigger than the efferent, and that helps maintain hydrostatic pressure, which is what drives the production of filtrate. Basically, if the door out is tinier than the door in, pressure is going to build up. And the whole point of that is to keep making urine. Yeah, you can come in. If the hydrostatic pressure were to drop significantly inside the glomerulus, we would quit making filtrate. If we quit making filtrate, that means we're not producing urine. If we don't produce urine, then nitrogenous waste and toxins are going to begin to build up in the blood. This is why one of the things that happens when you have a massive hemorrhage is eventually you go into kidney failure, because you lose that hydrostatic push that you need, that's driven by the pumping action of the heart and maintained to some extent by this difference in the diameter between the afferent and efferent arteriole that leads into and out of the glomerulus in the nephrons of the kidney. One of the things that the kidney does in order to maintain its blood pressure is that it can widen the afferent and narrow the efferent, or vice versa, in an attempt to try and maintain a relatively steady pressure with transient changes in the body's blood pressure. But again, that only works up to a point. And then what you've got here in between the afferent and efferent arteriole is the distal convoluted tubule. Okay, and what's critical about that is that there are cells that sort of surround the distal convoluted tubule and the afferent and efferent arteriole that make up something called the juxtaglomerular apparatus. And what it does is it regulates the diameters of the afferent and efferent arterioles based on the osmolarity of the filtrate that's in the distal convoluted tubule, so that we maintain a relatively constant blood pressure and electrolyte balance. And what you're seeing here is just a diagram showing you what's happening in each part of the glomerulus. So this is just essentially showing you where filtration is taking place, where reabsorption is occurring. Okay, again, this is primarily in the PCT, and where secretion is taking place, which is in the ascending or in the distal convoluted tubule, and also to some extent in the ascending limb of the loop of Henle. Okay, this is sort of a simplification, but basically these are the three processes that occur in the different regions of the nephron. And you get a little bit more detailed look here in the next slide at what's happening exactly in each part of the kidney. Just remember, when we say reabsorption, we mean that we're pulling stuff out of the filtrate and putting it back into the blood. When we say secretion, we're taking stuff out of the blood or the surrounding tissue fluid and putting it into the filtrate, which eventually should end up in the urine. And this diagram is just showing you what's happening in the different parts of the nephron. So this is a great diagram, I think, to star and study, because this boils down really what's happening in the cortex and in the medulla and collecting duct as the filtrate flows through the nephron. So you can see here that the filtration is occurring at the glomerulus. Here you can see in the proximal convoluted tubule that we're picking up a lot of water and salt and a lot of useful solutes, glucose, amino acids, fatty acids, and so on. There is some secretion that happens here. Certain toxins get secreted into the proximal convoluted tubule, as do protons. And then you can see here in the descending limb that it's water permeable, and so as the filtrate dips down into the medulla, the osmolarity increases. And then by the time we head into the ascending limb, we now are actively pumping sodium chloride into the medulla to maintain the salt gradient and also to reduce the osmolarity of the filtrate. And then in the distal tubule, we secrete toxins and potassium and certain drugs, and we reabsorb a lot of water and salt. And then in the collecting duct, it's primarily an adjustment of the salt and water concentration. There is some potassium secretion and some urea and water reabsorption all along the collecting duct. But by the time we empty into the minor calyx, the final concentration of the urine has been established. Now, the urinary bladder is just a stretchable sac, and it's lined with transitional epithelium, and it fills with urine via the ureters that conduct it down from the kidney. The opening where the two ureters come in and the urethra empties into the bladder is called the trigony. And the reason we make a big deal about the trigony is that this is very often where UTIs, urinary tract infections, will set up in the urinary system, especially in women, because the urethra in women is significantly shorter than the urethra in men, which means that the trip the bacteria take isn't nearly as extensive. It's easier for them to gain a port of entry through a very short trip into the bladder than it is to make it through the three regions of the male urethra: the penile, the membranous, and the prostatic. You can also see that the exit out of the bladder through the urethra is surrounded by two sets of sphincters. There's an internal urethral sphincter, which is smooth muscle, and an external urethral sphincter, which we control, which is skeletal muscle. And so by the time the bladder gets a little over half full, even the most ardent use of the external sphincter will not stop urine flow. And this is a built-in reflex on the part of the body, because we don't want the bladder to fill up with urine to the point where it backs up into the ureters and into the kidney, because then you can damage nephrons, damage kidney function, and it could, over time, send you into kidney failure. So this is the body's way of making essentially an emergency evacuation to maintain the integrity of the urinary system and keep you alive. Here you can see the female urinary structures. This is a sagittal section through the pelvic region. So this is posterior, this is anterior, and you can see here the urethra, the bladder. This is the uterus, the cervix, the vagina. Okay, there is the ovary and the fallopian tubes, and there, of course, is the anus, the rectum, and the last portion of the sigmoid colon right up here. And this would be the sacrum leading down into the coccyx. Here you can see the labia majora and minora, which essentially surround the opening of the urethra and the vagina. And here is the male urinary pelvic region. And what we can see going from the outside in are several structures, the glands or head of the penis. Okay, this is the corpora cavernosa. This is erectile tissue. The corpora spongiosum, which is also erectile tissue. And there's the pubic symphysis. The three parts of the urethra can be seen here. This is the penile or spongy urethra. Okay, this leads into the membranous urethra right here. This is the prostatic urethra that leads directly into the urinary bladder. This is the prostate gland. This is the seminal vesicle. This is the bulbourethral gland. And this portion of the penis right here is called the bulb of the penis, hence the name for the bulbourethral gland near the bulb of the penis and the urethra. Other structures include the testes, which sit inside the scrotum. These coil tubes are called seminiferous tubules, and this is called the epididymis. This is where the sperm learn how to swim, and then the tube that connects the epididymis to the urethra is called the vas deferens, and we'll learn more about that in the presentation on the reproductive system. Here you can see the urinary bladder filling up. The bladder fills and then empties when you feel the urge to urinate. You can see here the bladder as it's stretching out. The wrinkles, the rugae go away in order to accommodate the increased fluid volume. This is the superior wall of the empty bladder. This is the superior wall of the distended bladder. And by the time you get a little over half full, you feel the need to urinate, and you may in fact urinate involuntarily. You're expected to avoid about 2 liters of water daily. Remember that we have a total body water volume of approximately 40 liters, and that accounts for your intracellular plus your extracellular fluid. Your intracellular fluid making up about 25 liters, and the extracellular fluid making up about 15 liters. Now, the intracellular fluid is essentially the water that's inside the cells, and it's about 40% of your body weight, while the extracellular fluid is about 15 liters, and it's approximately 20% of your body weight. And of that 15 liters, 12 liters is what we call interstitial fluid. That's fluid between the cells. And the rest, the other 3 liters, is made up of blood and lymph, okay? That being the blood plasma and the liquid component of the lymph. Now, urine output in a hospital situation is generally measured by a patient who's been catheterized, and the catheter has a tube that leads into a bag that will measure the amount of urine the patient is generating. The danger, of course, is that if we produce less than 2 liters, considerably less than 2 liters, we worry about perhaps renal failure, okay, or at least renal insufficiency. And so it's important for the clinician to monitor urine output in the event that the patient is unable to get out of bed and urinate in a container. At the level of tissues, multiple systems are going to deliver and remove these substances. So water comes in and escapes through different routes. The only way that water should be able to get into the body is either if it's inhaled, such as in human air, or if it's ingested. Other routes into the body aren't available. But when we talk about um, losing water, we can either do that through the urinary system, through your breath, through your feces, or through your sweat or your cutaneous transpiration. And so the idea behind that is that generally water intake should equal water output when you take into account all these different systems. Now this diagram is also showing you other components that enter and leave the body through these different systems. Remember that oxygen is picked up by the respiratory system and delivered via the blood to the cells. You can see that here, where carbon dioxide is produced by cellular metabolism and released when we expire. Nutrients, of course, are absorbed across the lining of the stomach and the small and large intestine, mostly the small intestine, while water and ions move between the cells and into the cells, and also between the cells and the blood and between the cells and the lymph. Okay, so this is a series of fluid compartments in the body that freely exchange material. Here you can see that ions and water are also exchanged between the kidneys and the interstitial fluid, but nitrogenous wastes are primarily eliminated as the blood is cleaned by the kidneys when it comes in through the renal artery. What we're looking at here is just the distribution of water coming in and going out of the body. We generate we produce, as a result of metabolism, about 10% of our body water, foods another 30% coming in, while beverages make up the remainder. And we lose water primarily in the urine at 60%, and then we have loss through our breath and through our skin, another 28%, sweat, the loss that we're aware of, another 8%, and feces, approximately 4%. Now, those percentages can vary a little, but they're basically fairly constant. If water input equals water output, then we remain relatively happy and healthy. If water input exceeds water output on a daily basis, then we run the risk of hypotonic hydration, which can disturb electrolyte imbalance and perturb cell function. And if we take in less water than we lose, then we run the risk of dehydration, which can also perturb electrolyte imbalance and disturb cell function. What you're looking at here is just one of the ways that we control electrolyte and fluid content in the body, and one of the major ways that this occurs is through checking the chemical composition of the blood with special sensors that are stationed throughout the arterial system. So if we take an example of somebody that has a normal fluid balance and then they experience an increase in plasma solutes, which means that the osmolarity of their plasma goes up. Osmoreceptors in the hypothalamus, as well as chemoreceptors in the in other areas of the body, in the arterial system, will signal the brain to stimulate the thirst center. This produces a sensation of thirst, and we seek out liquid. The water moistens the mouth and the throat. It stretches out the stomach and the intestine, and that gives us a temporary satiation. But what happens as a result of drinking water, of course, is that as we take in water and we dilute electrolytes, we reduce the concentration of plasma solutes. That in turn causes the osmoreceptors in the hypothalamus to quit signaling, and it also cuts the sensation of thirst, and as a result everything goes back to homeostasis. In addition, as we increase plasma solute concentration and thus osmolarity, we see a decrease in saliva production, which produces dry mouth. That also has an input on the hypothalamus, which causes us to seek out moisture, which again moistens and cools the mouth, distends the intestines, and of course is absorbed into the bloodstream and ultimately that signals back to the osmoreceptors in the hypothalamus, but also gives us a short-term satiation in terms of fluid intake. Okay, now one of the things that we have to understand is that the kidneys are vital in maintaining blood volume and blood pressure and electrolyte balance. And what you're looking at here is a diagram that illustrates what we call the renin-angiotensin-aldosterone system. And it's triggered essentially by an increase in blood osmolarity, stimulation of thirst, and a drop in blood volume. And the idea here is to restore blood volume and blood pressure. So the way this operates is that the liver is always making a hormone precursor called angiotensinogen, which is present in the blood. When the kidneys sense a high plasma potassium level or are stimulated by ACTH that comes, of course, from the anterior lobe of the pituitary, or if they are triggered via result of essentially low blood volume or high blood osmolarity, what's going to happen is they're going to produce now a hormone called renin. And what renin does is it converts angiotensinogen into angiotensin 1. And in the lungs, what happens is there's an enzyme called angiotensin converting enzyme that will convert angiotensin 1 to angiotensin 2. And from there, what will happen is that the effects of angiotensin 2 will be to increase the sense of thirst, to stimulate the production of antidiuretic hormone, and to vasoconstrict the blood vessels. And so what does all this do? Well, the vasoconstriction of the blood vessels will help to raise blood pressure. Thirst will cause us to seek out water to increase blood volume and, as a result, blood pressure. And of course, antidiuretic hormone will cause the collecting duct to become more permeable to water so that we retain more water from the urine, thus increasing blood volume and blood pressure. Okay. Now, at the same time, the ACTH that's going to be released by the hypothalamus is not only going to trigger the release of renin from the kidney, but it's also going to stimulate the adrenal cortex to release aldosterone, which is your salt-retaining hormone. And the result of that is that the kidneys will absorb more sodium and secrete more potassium, the result being that there will be more sodium retained but also more water retained, and as a result blood pressure and blood volume will go up. Okay, so these are just a few of the things, of course, to keep in mind. Now, once we restore fluid volume and blood pressure and circulating blood volume, what will happen is it will have an inhibitory effect on the renin. There won't be any more angiotensin 1 made, which means that angiotensin 2 levels will begin to drop. That in turn will act to vasodilate the blood vessels. In addition, we won't be stimulating the hypothalamus via thirst or ADH to produce adrenocorticotrophic hormone, and the whole system will go back to normal. Okay, so it's a little bit complicated, but essentially the initial stimulus is a drop in blood pressure or an increase in blood osmolarity, and that's what gets the whole ball rolling. Okay. Now, what you're looking at here is just a diagram showing you how it is that the kidneys are able to maintain the pH of the tissue fluid and also of the blood plasma. Bicarbonate is reabsorbed, and this decreases the number of protons in the blood, and what that does is it makes the blood more basic, because what happens is that the bicarbonate will neutralize the free protons, forming carbonic acid, which is a weak acid, so it doesn't dissociate into free protons as easily, and as a result the pH will begin to rise. Bicarbonate is excreted, which can increase the number of protons in the blood, causing it to become more acidic. And so what the kidneys can do is either excrete or retain bicarbonate, or they can excrete or retain protons. They can do either one. So what you can see here is just an example of how this affects the urine. Fewer protons results in the blood, results in less bicarbonate in the urine. More protons in the blood um, results in, I'm sorry about that, more protons in the blood is going to result in um, more bicarbonate being resorbed into the plasma, being taken out of the urine. Less protons in the blood, the more basic the blood, what's going to happen is we're going to secrete bicarbonate, and the result is that the blood pH is going to begin to drop. Okay, so whatever happens in the blood, the urine is going to undergo a chemical readjustment by the kidneys, such that if your blood pH or your tissue fluid pH were to drop, your urine pH would drop, and if your blood or tissue pH were to rise, your urine pH would rise, and the result would be, again, now a stabilization of pH. What you're looking at here is just a microscopy of the kidney, and what we need to understand about the kidney is that it is made up of millions of filtration units called nephrons. Remember that we've got four main regions of the nephron. You've got the Bowman's capsule, which you can see here surrounding the glomerulus in the kidney. The proximal convoluted tubule, you can see some cross sections through PCTs here as well. The loop of Henle, which dominates the medulla, which isn't really visible in either of these pictures. And then the distal convoluted tubules, which are also going to be found in the cortex. And then finally the collecting duct, which isn't visible in either of these slides and is also going to be found in the medulla. Remember that the reason that the pyramids, which are found in the medulla, have that striped appearance is because of the parallel arrangement of collecting ducts, loops of Henle, and also peritubular capillaries. Now, make sure that you're able to recognize the simple cuboidal epithelial cells that are the hallmark of the distal and proximal convoluted tubules, and make sure that you can pick out a glomerulus. This is the capsular space, and this is the Bowman's capsule when you see that on a diagram, say on a PowerPoint. You want to make sure that you understand the flow of blood through the kidney and the flow of filtrate through the nephron and out of the kidney, and the flow of urine. And so what you're looking at here, again, remember, is cross-section of the kidney. This is, of course, the adrenal cortex. This is the adrenal medulla. This is the capsule of the kidney. This is the cortex of the kidney. Okay, these are the renal columns. All right, these are the renal pyramids, and these are the minor calyces. That's a major calyx where two minor calyces converge. There's another one. There's another one. Renal pelvis, ureter. This is the renal artery. Remember that the blood flow goes from renal artery to segmental arteries, to interlobar arteries, to arcuate arteries, to cortical radiate or interlobular arteries, to afferent arteriole, to glomerulus, to efferent arteriole, to peritubular capillaries, and then we head back out of the kidney via the cortical radiate veins, to the arcuate veins, to the interlobar veins, ultimately to the renal vein, from the interlobar veins to the renal vein, and from the renal vein back to the inferior vena cava, and of course from there back up to the heart. Okay, so that's the blood flow. For the flow of urine, remember that we form the urine initially in the glomerulus at the Bowman's capsule. It flows from there to the proximal convoluted tubule, from there to the descending limb of the loop of Henle, which dips down into the medulla, from there to the ascending limb of the loop of Henle, which heads back up towards the cortex, and then to the distal convoluted tubule, from there to the collecting duct, and from the collecting duct we're going to empty through the renal papilla into the minor calyx to the major calyx. The urine will then enter the renal pelvis to the ureter, then it'll head to the bladder. From the bladder it'll head to the urethra, and that's the end of the trip. This is just a look at the kidney for identification purposes, basically laying on its side. And this is just the kidney model we were looking at earlier. Remember to be able to identify all the different parts, right? The glomerulus, there's the PCT, there's the descending limb of the loop, the ascending limb of the loop, the DCT, the collecting duct. This is the renal papilla. This is the minor calyx. Okay, so to review, glomerulus to Bowman's capsule to PCT to descending limb to ascending limb to DCT to collecting duct, from collecting duct through the renal papilla into the minor calyx to the major calyx to the renal pelvis to the ureter to the bladder and then to the urethra, and then that's the end of the trip. Remember here that you're looking at a Bowman's capsule. This is the glomerulus, this is the afferent and efferent arteriole, this is the distal convoluted tubule, this is the proximal convoluted tubule. Okay, remember that the filtration membrane for the glomerulus has 3 layers. You've got the endothelial cells of the capillaries, then the basement membrane, and then of course these white cells called podocytes that surround the outside of the capillary. And what this does for filtration is it only allows molecules below a certain size to get into the filtrate. Also remember which processes dominate each part of the nephron. Filtration, of course, happens between glomerulus and Bowman's capsule, and then primarily absorption in the proximal convoluted tubule, where we rescue water, electrolytes, and useful solutes such as amino acids and glucose and fatty acids and put them back into the peritubular capillaries. In the descending limb of the loop of Henle, we primarily reabsorb water out of the filtrate and into the kidney. In the ascending limb of the loop of Henle, we pump sodium and chloride out of the filtrate and into the kidney. That's what establishes the salt gradient in the medulla. And then in the distal convoluted tubule, we primarily secrete toxins and nitrogenous wastes. And then the last trip through the collecting duct is where we adjust the water content of the urine, and that's essentially where the composition of the urine, once it leaves the collecting duct, no longer changes. From the time we enter the end of the collecting duct all the way through coming out the end of the ureter, the composition of the urine is essentially unchanged. Okay, that brings this portion of lab discussion to an end. Remember to study this material. It will be covered on next week's practical and final exam. Thank you.